All right, guys, I got a lot to cover today, so I'm going to jump right into it. We're going to break up chapter 19 into two chunks. The second chunk is going to be a little bit less cumbersome. There'll be a little bit less material in it. But today I'm gonna talk and talk and talk and talk, and you're going to be confused and confused and confused. So um, if you are confused about this stuff, remember we will have our next open office hours next Monday from 1:15 to 1:45. Um, this chapter expands on the genome of eukaryotes. Kind of gets into detail about um, some of the things that we've been discussing in chapter 16 and 17, which are what the DNA does, how it replicates, how it transcribes, translates, creates proteins, things of that nature. And today we're going to get into how the genome, the DNA, is organized into genes, what the different components of genes are, even though you do have an inkling of what that is already a little bit. Some of the stuff will sound familiar in that respect. Um, and we're going to talk about how we regulate the expression of genes as well. So all that stuff really is quite related to the stuff that we've been talking about. So without further ado, um, start here on this slide. It says three features of eukaryotic genomes are a major information processing challenge. The reason being is that they're much larger. A lot of the stuff that scientists look at when they study our genome is how it compares to bacterial genomes. Bacterial genomes are really, really simple, easy things to study. They're relatively small. They have very few genes. Um, and most of the genes get expressed most of the time. Um, for us, that's difficult because our genomes are much larger, obviously. There's limited expression of many of the genes in specific cells, right? So if you have a eyeball cell, only genes that code for eyeball stuff are going to be on, and that's not necessarily the case in bacteria. And unlike bacteria, our chromosomes, as it states there, our chromatin is highly ordered into higher structural levels. So what I want to do first is kind of just talk about how our DNA is packaged, and some of this should be reviewed. We've talked about this before. We've seen this before. We've seen an animation that puts this all into perspective. But I do want you to understand that at some level, the extent to which we package our DNA into these chromosomes inhibits protein creation. Okay, it inhibits transcription and ultimately translation. So I'd like to talk about that real quickly and discuss how our DNA is packaged, how it is folded into more condensed chromosomes. And when we talk about this folding into these condensed chromosomes, those things that look like butterflies that we see in the pictures all the time, those are put together for convenience. Actually, very little time is actually spent in that, but it does take some unpacking if you want to go ahead and, pardon me, if you want to go ahead and open that DNA up to actually transcribe it and get some kind of protein from it. So the packaging starts at this DNA double helix level. This is unpackaged, unfolded DNA, okay, DNA double helix. Now, what we can do with that is we take that DNA double helix and we start to wrap it around something called histones. These histones are going to play a role in the regulation of gene expression as we work our way through some of this information. So they are going to become important. Okay, so these little purple beads that look like they have little tails on them are called histones, and they are little proteins, and they work like spools of thread, right? They would be like the little plastic thing that you wrap the thread around, and the thread would be the DNA. And so if we look here, we can see these little blue strands of DNA wrapped around them, and that's our first level of protein folding. It allows the DNA to kind of be collected all in one spot. And here over on the microscope view, we can see these little beads. These little beads are called nucleosomes. Nucleosome is the term that describes the histone and the DNA together. Okay, so scientists name everything, and that's what they've named that one. So that's what we see there. Then we take these nucleosomes, the little histone DNA packages, and we tighten them up. Okay, this is what scientists call the chromatin fiber. Okay, the chromatin fiber, and here we can see it in the microscope on this side, but what that looks like ideally is that you get these little nucleosomes all packaged up real nice. You see how organized that is? It's as if somebody, actually took this thing and specifically folded them and placed them next to each other so they'd all fit as tightly packaged and ordered as they possibly could. From there, we attach these little chromatin fibers, which we saw in the previous one, to this protein scaffold. It's basically just like a dock, and it doesn't do anything. It's just a piece of protein. And basically what we've done here is we've taken this chromatin fiber and we've kind of looped it and we've attached it like little anchor points right down here. And so what this does allows us to kind of organize this DNA very nicely, put it in a tightly ordered structure here with these little loops, and we can attach them closer and closer together. Okay, these are called looped domains. And as you can see, what's happening is that these, if you are paying attention to the scales here, the scales are getting bigger. Okay? In the previous one, it was 30 nanometers thick. Now we're at 300 nanometers thick. So what we're doing is we're increasing the thickness of these things as we package it all together. Okay. Then we take those loop domains, what we saw in the previous, okay, this thing right here, this picture right here. We're going to take that and we're going to coil them up even more. We're going to fold them up, package them up, smash them together even more. Kind of a neat little thing. And then what we do is we shove all those together into this chromosome, which ultimately becomes about 1400 nanometers thick. Okay, so we start, I think the DNA itself, the DNA double helix is 2 nanometers thick. And by the end of all this packaging and protein folding, we end up getting a structure that's 1400 nanometers thick. So we increase the thickness of this DNA package very extensively. Okay. So the loop domains coil and fold and they form the metaphase chromosome. All right, now that's how we put all the DNA actually together in a single spot, usually, or I shouldn't say usually, but oftentimes that DNA is found uncoiled. It's found in this conformation right here. That's when we can actually utilize the DNA. Once we start packaging it up into this chromatin fiber, it's unusable. You see all that DNA in there. There's no way that you can access any of the base pairs to actually read them. So at the level of chromatin fiber folding, that DNA is inaccessible for transcription and translation, for protein folding, or for protein creation. Now, when we get down to the actual double helix DNA, we do have to understand what's going on organizationally there. So we're kind of zooming back in to very, very small scales here. And this is oftentimes how genes or genomes or areas of interest in a DNA strand are represented in our book. And oftentimes if you look up pictures of, blah, blah, blah gene, whatever gene you might be looking for, it might give you some kind of representation like you're seeing here, okay? But I want you to have an understanding of what you're looking at here because we're going to see this particular type of thing over and over and over again in the rest of this PowerPoint. So I want to make sure you're paying attention to this and just kind of give you a brief introduction. We're going to come back. This exact same picture is in another slide later on, but I want to give you a brief introduction as to what this is. At the top here, we see our DNA. Okay, the dark stuff that we see right here in the middle, that is the gene of interest. That's we call that the transcription unit. That is a term that we used in chapter 17. This is your transcription unit. This is the gene or the stretch of DNA that is going to be transcribed. Okay, now this is our zone of interest. Okay, and so from there, that zone of interest, we can start naming things in relation to that. Okay, now up here, we call this upstream, okay? Upstream. It's upstream because the start of the gene is here, okay? We are upstream of that, okay? If you think about utilizing the term upstream in terms of like rowing or being on a river or something like that. I'm going to use a gross analogy here to help you kind of remember that this is called upstream. Let's say you're sitting right here, okay, in the stream. You're just kind of laying there. It's nice out, nice weather, and you're kind of cooling off, and you got a bunch of wise guy friends upstream, okay, and they think it'd be hilarious to go pee in the river right here, okay? The pee is going to go downstream to where you're at, which is at the starting point of this gene near the promoter, right? So it's in this scenario, you would rather be upstream with them because you don't want to get peed on. Okay, that pee is going to go downstream. Totally off the wall, totally makes no sense in any other context, but hopefully it works for you. Okay, so it gives us some indication as to what is considered upstream and what is considered downstream. Okay, if your friends are upstream from where you're at, the pee that they put into the river is going to reach you as it goes downstream. Okay, hopefully that works. If not, skip over that entire analogy. We see the promoter. We've talked about the promoter before. We see the terminator. We've talked about the terminator before. Those are two things that we've discussed in chapter 17. We see these dark blue things called exons. We see these light blue things called introns. Again, both chapter 17 things, which we've talked about, and they'll come back into play here. But these up here are new. These enhancers or control elements, they're called, okay? These yellow things are called control elements, and those are portions of DNA that actually go with, if you want to say that, go with this gene. They are instrumental in making sure that this dark blue gene right here actually gets transcribed, okay? Now, what we see down here is a representation of the mRNA transcript that is created from this transcription unit. And then down here, we see the modified mRNA transcript. Okay, so it takes this one, cuts the introns out, adds the poly A tail and the 5' cap. So a lot of the stuff you see in this picture is just a different representation or a review of some of the stuff that we've seen in chapter 17. All right, let's go ahead and get to the meat of what we want to cover here. The bulk of this chapter is going to deal with how our bodies, how our cells regulate the transcription of genes. Regulate, and not necessarily just the transcription, I should say regulate the expression of genes, okay? The regulation of gene expression is absolutely important to how our bodies function because it allows us to fine-tune how much stuff we are creating, how many protein products we're creating. It allows us to fine-tune, or not fine-tune, but shut off translation, allows us to shut off a gene if we need to. And that's what we're talking about here. Now, this kind of goes back in and relates to the end of Unit 4 as well when we talked about differential gene expression, because this process we're going to talk about here, how we regulate these genes, yields or causes differential gene expression. It causes these differences in the cells that we see in our body. It causes the differences that we see structurally between, let's say, liver cells and heart cells, or skin cells and eyeball cells. All of those differences structurally are because certain genes are shut off or regulated in certain areas of the body. And so when we're studying some of this stuff, try to think back to what we were discussing when we said, okay, in certain parts of the body, certain genes are turned off while certain genes are turned on, or maybe a certain gene has a very specific rate of transcription or rate of translation, okay? Now, when we are discussing this phenomenon of gene regulation, gene expression regulation, we have to remember what that term gene expression means. The term gene expression means that you're turning on a gene, you are expressing a gene. You're ultimately, when you express a gene, the idea is that you get some kind of protein product at the end of it, right? Now, when we talk about gene expression, there is a lot that goes on with gene expression. Everything from accessing the DNA, okay, what we see up here, this raw DNA, we've got to access it. Everything from accessing the DNA to transcribing it, to processing the RNA, to getting it out of the nucleus, to translating it, and then we've got to modify the protein, and then we've got to turn the protein on, and then we've got to get the protein to the right spot. All of these things are considered gene expression. They are involved in the process of gene expression. So this entire diagram that you see right in front of you here is gene expression. I want you to keep that in mind. And your cell can basically regulate gene expression at any one of these stages right here. We're going to talk about so many different ways that you can regulate gene expression, and we're going to talk about how you can regulate it during transcription. You're going to talk about how you can regulate it just by accessing the DNA. How do you regulate RNA processing? How do you regulate translation? How, what goes into that? Okay. So now, when we discuss regulation, it doesn't necessarily mean just turning on or turning off genes. There are subtle things that can occur when you are all along this path that will change either how much of this protein you're going to get, or whether or not you're going to get it at all, or what version of that protein it is. Okay, but do make sure that when we continue throughout the course of the next however many slides we cover here, we are going to talk about ways of regulation of gene expression at every one of these levels, just about. Okay, so let's jump right into it. We can regulate gene expression by chromosome structure. Okay, I talked about at the very beginning here the fact that chromosome structure, DNA structure, how it's packaged up is vitally important to being able to access the DNA so that you can transcribe it, right? When we look at the very base level of DNA folding, we talk about histones, right? It was the histones, and this is almost an identical picture to what we saw before. We talk about the histones, these little guys right here, these little colored spheres with the tails on them. Those are histones, right? And we have the DNA that wraps around them, like the spool with the thread. Now, what happens to these histones is that they can become acetylated. Okay, it's a kind of a big word. You don't really necessarily need to know what it means, but it means that you are adding acetyl groups, which are organic compound groups, to the tails of these histone molecules. Okay, so we can acetylate the histone groups, and what that does is that's going to loosen up the DNA so that you can actually access it. Now, this picture, I wish it was bigger, but it's attached to this other picture. This picture down here, I think, is the best understanding of acetylated versus unacetylated histones and what they do to allow the DNA to be accessible. Okay. Now, if you look at this picture here, this is all of these little nucleosomes. That's what those are. These nucleosomes packaged up. This is the chromatin fiber. This DNA here is inaccessible. We cannot transcribe this DNA at all. It's too packaged up. We can't get our transcription machinery in there to actually access it. But what we can do is we can access it. There is a way to get at the DNA. And one thing you'll kind of point your attention to here is that you look at these little purple tails that kind of stick up right here. Those are those histone tails that we see here. Okay? Those little histone tails do not have any little green spheres on them at this point, okay? But in this picture, they do. This picture, those little green spheres are acetyl groups, okay? And when we add these acetyl groups to those little histone tails, what it does is causes some kind of uh, repulsion force between these histones, and it causes them to be packaged up. They were packaged up like this, and then you get those groups on them, and then they go like this, okay? And then they move away from one another. So when we add those little green acetyl groups to these tails, what it does is it causes those nucleosomes, these little beads, to separate from one another. And when they separate, you will notice that there is now stretches of DNA. There's a stretch of DNA here, there's a stretch of DNA here, and here as well. That is now open for business. Okay, that is a little stretch of DNA that can now be transcribed. Okay, it's just double helix. That's all it is. And so if there's a gene in this little area here, all of our transcription machinery can get in there and transcribe it and make mRNA from it. Okay. So histone acetylation allows us to regulate the process of gene expression by actually accessing the DNA, making it accessible. Okay. So this process seems to loosen the chromosome structure and promotes the initiation of transcription, allows transcription to actually start. Okay. There is an opposite process known as DNA methylation, which is almost the complete opposite process of DNA or histone acetylation. DNA methylation is the addition of methyl groups to certain bases in DNA. And so what occurs here is that when we get these methyl groups, and again, it's an organic compound that's added to the certain bases. I think in the slide I'm going to show you here, they are added to the cytosines. Okay, and when we get those things added, it almost takes that structure that we saw blown up like that, as all the stuff was separated, it brings it back down and tightens it all up again. Okay, and it almost locks it into place. Now, in some species, the methylation can cause long-term inactivation of genes, and this leads to something known as epigenetic inheritance. And I've mentioned epigenetic inheritance before. And your body, your DNA can actually pass down DNA methylation to the next generation. And so you don't oftentimes get that type of thing passed down in generations, or we hadn't known about it before. Okay, so this picture up here shows us, I like this one, I think probably best when trying to illustrate DNA methylation. This is a stretch of DNA that has been expanded. It's probably got, the histones have been acetylated, right? It is basically this picture right here. Okay, so this picture and this picture right here are basically the same. And when we add these little red methyl groups, that's what we see here. When we add these little red methyl groups to the DNA, what it does is it causes these guys right here to kind of ball back up. Okay, you see how they're kind of expanded and then they crunch back down, and then they actually get locked in place like that. So what this does is this causes that DNA, there's no longer a stretch of DNA. So this stretch right here could be transcribed. There's plenty of room for transcription machinery to work here. Now all that stuff is locked back together, okay? And these DNA methylation patterns can actually be sent down and inherited by the next generation to your kids, actually. All right. I'm going to read the top bullet point word for word. It says, once a gene is modified for expression, meaning if you look at a gene and you've modified it for expression, you're opening it up, okay? Usually through acetylation. Transcription initiation is the most widely used stage for gene regulation, okay? So what this statement is telling us is that we can, or the cells, regulate transcription initiation. And this means that the cell is regulating the start of transcription. That's what transcription initiation is. The cell is going to do different things to regulate whether or not transcription actually begins. Okay, so to study this mode of regulation, and again, this is the most widely used type of regulation because there's so many different things that you can do with it. And it's the stage that actually makes it so you have to turn the gene on. There's different forms of regulation that can happen after transcription's occurred, but once we make that mRNA, oftentimes we're just going to make the protein. So we want to be able to tell the cell, do we even need to make that mRNA at all? Okay, and so that's why this is probably the most widely used and maybe the most important mode of regulation. So to study this mode of regulation, we need to delve deeper into the organization of the eukaryotic genome, which we'll do. I'll expand a little bit more on the organization of this genome. The role that transcription factors play. We have yet to really discuss in depth and detail what transcription factors do. We've mentioned them, we haven't talked detailed enough about them yet. And then the role of what enhancers do, as well as specific transcription factors. Okay, so here's a little bit more on the organization of that genome, the genome. I've gone through a lot of this already. This is what I went through in the very beginning portions of this PowerPoint. But I want to center your attention a little bit more closely on these things that are found upstream, okay? The control elements. We've got two different types of control elements. We've got what are called the proximal control elements, and the proximal ones are located closer to the transcription unit, closer to the gene. Okay, that term proximal means closer to or closer, and that's exactly what we see here. Okay, this one here, the distal control elements are located further away from the gene or further upstream from where the gene's at. These have an alternate name that they go by called the enhancers. Okay, so the enhancer is the distal control element, the ones that are located here, and these are little sequences of base pairs that we see here in yellow. Okay, but the control elements are segments of non-coding DNA, which means they don't really do anything. They do something, obviously. We're going to talk exactly about what they do. They don't actually give you proteins. That's what's meant by non-coding. They don't code for proteins, okay? But they do have an extremely important function, which we'll talk about. And they do help regulate transcription because they bind certain proteins. And when we see these things bind certain proteins, and I'll show you, or I'll link, it's kind of hard to visualize this stuff when I'm just talking about it. So I'm going to also link a video that shows you these things in action. Hopefully that kind of sheds some light on it. Okay. So the control elements are critical to the precise regulation of gene expression in different cell types. So these control elements, even though they're non-coding and even though they don't give you mRNA or genes or anything like, or proteins or anything like that, transcription can't happen without it because it is like the turn on button. It is like the go button. It is like the key that unlocks the gene. Okay, so we're going to talk about how those work. But to do so, we need to discuss what transcription factors are. Okay, we've mentioned these before. I have mentioned these in chapter 17, the fact that they're needed to start transcription. But we're going to talk about how transcription factors work with those control elements that I just mentioned, the enhancer and the proximal control element. Okay, so to initiate transcription, RNA polymerase actually has to latch on to these things called transcription factors, and they're protein in nature, and they work kind of like little docking bays, and I use that term a lot, but that's to make sure that there's proper orientation of the RNA polymerase, to make sure that it gets latched onto the spot that it needs to be to accurately transcribe the gene that we want to make a protein out of. So these transcription factors are kind of structural proteins, scaffolding proteins that need to be there for RNA polymerase to do what it's supposed to do. So we have general transcription factors that are essential for transcription of all protein coding genes. And then depending on which gene you might be trying to get a expression from, you might be working with something known as specific transcription factors, and those are specific to certain types of genes, okay? But there are certain types of transcription factors that are needed for all protein coding genes, whereas some other specific genes, there are specific transcription factors. All right. So let's discuss proximal control elements are located close to the promoter. Remember, the promoter is where ultimately the RNA polymerase is going to bind to. It's kind of like the starting point for transcription. The distal control elements may be very far away from the gene. They could even be in an intron, okay? And then we've got these things we have yet to discuss called activators and repressors, okay? Activators and repressors work with these control elements. They bind directly to the control elements, okay? An activator is a protein that binds to an enhancer and stimulates transcription of a gene, and a repressor inhibits the expression of certain genes by blocking activators. They can actually sit in the enhancer region and kind of block the activators from doing what they're supposed to do, okay? So we've got two ways here of regulating gene expression. We can bind these activators to the enhancers, and when we do that, the gene will be transcribed. Or we could bind repressors to these enhancers, which causes the genes to not be transcribed. Okay, it represses transcription. All right, so this is kind of how they work. This is a sequence of events that starts up here with this picture. We notice the gene down here, downstream. We notice the promoter region along with the TATA box. Okay, that's basically going to be the starting point for RNA polymerase. And way upstream here, we notice the enhancer. Okay? The enhancer in this picture has these little activators. These are chemicals. Okay, there's going to be compounds, and they're going to bind to the enhancer. Now, when all of the enhancers are bound properly to the activators, what we notice here is that something is brought in. There's some kind of activation that occurs, some kind of change in the DNA structure that allows this thing right here to come in and do a little work. It's called a DNA bending protein. And what it does is it causes this DNA, it kind of sets up right here, causes DNA to bend back on itself. Okay, it bends back on itself. And you notice that these little activators right here they look like they want to do something important. Okay, and ultimately they will. These transcription factors now come in. These little things that look like bunches of grapes. These are all considered transcription factors, and they all come in. I call them scaffolding proteins. They kind of are. It's not a totally accurate representation of what they do or what they are, but they provide some scaffolding, some structure for this entire complex. So these transcription factors come in here and they bind to this area, the promoter. And once they do that, you notice that these activators have a very specific shape and they bind up to these little transcription factors. And we see that occur down here in this bottom picture. The little activators connected to the enhancers will then connect to the transcription factors. Okay? Once this has all kind of come together, we see RNA polymerase attached, this little translucent guitar pick. Once RNA polymerase attaches, we can actually have transcription of this gene. Okay, but all of this stuff has to happen properly and in the right order for RNA polymerase to even be able to have any understanding of where it's supposed to be. This giant complex of crap right here, this purple stuff, all this stuff, this giant blob right here is a beacon for RNA polymerase to come in and do the work that it has to do. Okay, so this is one way that we can turn on a gene. All right. Now, depending on which genes you're working with in which cells, remember that all of the cells that we have in our entire body all have the exact same DNA. They all have the exact same enhancers. They all have the exact same promoters. They all have the exact same terminator regions, genes, distal, proximal control elements. All of that stuff is the same. And so this picture here describes how we can actually turn on just certain genes using certain enhancers in certain cells. Okay, and that's at the very end of Unit 4. We talked about differential gene expression. This is what it is. This is what it's all about. This picture right here illustrates how the liver cell is going to express an albumin gene. Okay, you want albumin expressed in your liver. Okay, but you do not want the crystalline gene expressed in your liver. Then you'll start making eyeball parts. Okay, and the way this occurs is that the genes in your liver cells are going to utilize certain activators. And those activators are what are going to be available to only turn on a certain type of gene. Okay, now so that's what we see right here. In this picture here, the albumin gene in the liver cell, it's in the eyeball cell as well, but we're worrying about it in the liver cell, has an enhancer region that needs a red activator and at least a gray activator. Okay, and at least for this particular picture, it doesn't look like the yellow activator necessarily matters. Okay, but what we notice here is that in this picture, somehow there is a reason behind the somehow, but we're not going to get into what that is. These little red molecules here, these little red activators and these little gray activators are available, okay? And they will bind to this portion of the enhancer. The DNA will curve over back on itself, and it will bind to specific transcription factors right here in the promoter region, okay? And so when we get these very specific activators available in this very specific cell, it is this enhancer region that will bend back on itself and initiate transcription of this particular albumin gene. We notice that we only have one of the transcription factors here, okay, and this is probably a general, this gray one is probably a general transcription factor. This general transcription factor right here has bound up, but we don't have any of the other specific ones. There's just no more activators that are in this particular cell because this is a liver cell. It's not an eyeball cell. Now, if we look at the eyeball cell, okay, we look and see which activators are available here. Okay, we've got the pink one, we've got the orange one, and the gray one. We've got all three for this one. And so all three of these activators bind to the enhancer region. That enhancer region bends back on itself like we see has happened here. Those activators work with these transcription factors. They bind the RNA polymerase. Then the RNA polymerase can express the gene for crystalline. Okay, it is a very nice system that allows you to have every single cell in your body have the exact same genetic structure and genes available to it, while at the same time only activating certain ones. So if we see here, we've got everything the same in both cells, but we're only activating a specific gene in each type of cell because those are the activators, those are the chemical messages that we have available to this cell. There's a very good chance that these activators came from or are the result of induction by nearby cells during development. This liver cell very well could have gotten a signal from some other cell nearby that says, okay, you're going to produce albumin. So here are red and gray activators to do the job. Okay, so it's that's kind of how it works. All right. Let's see. I don't know how long I've been going here. We've only got a few more slides left to go, so I'm just going to plug on. This will be a really long one. The next one will be shorter, so bear with me. So initiation of transcription tends to be the most important stage. That if you heard that, that was not a fart. That was the van when I switch it to the air conditioning. Makes like a fart sound. So I did not fart. Anyway, the stuff that we just talked about, the regulation of transcription initiation tends to be the most important and most widely used regulation step in gene expression. But now we're going to talk about some of the others. I'm going to go way back here to this picture. Talked about all these different methods or ways that we can regulate gene expression. Now we're going to start to delve into some of the other ones. We've just talked about regulation of transcription initiation, which is basically this step here where the little red dots at. Okay. Now we're going to get into all these other ones, but it's not going to take us a lot of time because we're not going to go into a lot of depth on them. This is probably the one we'll go into most depth about, that we can regulate gene expression by regulating post-transcription. Okay, so if you recall, one of the things that we have to do before we get an actual functioning mRNA strand is we've got to modify it. We've got to add some guanine caps, we've got to add some poly A tails, we've got to get it out of the nucleus, we've got to cut out the introns and things of that nature. We can regulate a lot of that processing, okay, and that's what we'll talk about here. So post-transcriptional regulation allows the cell to fine-tune gene expression rapidly in response to environmental changes. So if there's something that happens to have changed very, very quickly, even after you've transcribed your gene, this processing or this regulation allows you to adapt to that particular instance. Okay. So there's 4 ways in which we can regulate after transcription, which is what post-transcriptional means, right? We can regulate RNA processing. We can regulate mRNA degradation. We can regulate the initiation of translation. We can regulate protein processing, and we can even break down proteins that we might have too many of. All right. So one of the things that we can regulate in RNA processing is we can regulate how we splice things. And I think I alluded to this in chapter 17. Chapter 17, yeah, chapter 17, when I talked about splicing, that you can alternatively splice your RNA, your mRNA molecules, to yield two different mRNA products. Okay, and this is based on the introns. Depending on the situation at hand, you can determine which stretches of RNA are exons and which are introns. In fact, you can actually take some introns and turn them into exons, and some exons and turn them into introns. And so you can flip things around, you can shuffle things around. It offers great variability in the amount of mRNA and protein products that you can create just by switching these things around, just by determining which exons are exons and which introns are going to be introns. And so what we see here is a picture of the DNA that we're transcribing. These are all the exons in the different colors. Creates this mRNA, this RNA transcript, and depending on how we see it, depending on how we cut it, we can get two different mRNA products, which are going to yield two totally different proteins. Okay, we get this one on the left. You can't see it now. This one on the left has a little green chunk in it, and the one on the right has a little purple chunk in it. Those are two different products. They're all exons, but you can determine which ones are going to stick around and which ones aren't. Okay, so we can modify or regulate how we process the RNA. That's an effective way of regulating gene expression, right? Not necessarily shutting it off or turning it on. We're fine-tuning the expression. Okay, so that's a different way of looking at it. mRNA degradation. I like the way they put this here. The lifespan of mRNA molecules in the cytoplasm is key to determining the amount of protein that is produced. If the mRNA sticks around longer in the cytoplasm, you would naturally understand that you're going to get more protein. If the mRNA is around longer, it's going to be translated more often. Okay. Now, if we don't need a bunch of the protein, we don't want that mRNA to stick around a long time, right? We want to get rid of it. So there are effective ways, or ways that the cell effectively can shut down the mRNA once it's been created. Let's say the cell realizes, oh my gosh, we've got way too much of this protein now. We need to stop protein production. How do we do that? Well, we can block the mRNA so nothing can attach to it, so you can get no ribosomes attached to it at all. Or we can just chew up the RNA. We can just chew it up and get rid of it, kill it, okay? And so this RNA interference is done by something known as microRNAs. Okay, they can lead to the degradation or the breakdown of an mRNA, or they can block the translation altogether. Okay, so that's kind of what we see going on here. This stuff over here, you don't necessarily need to worry about. This just talks about how the miRNAs work, the microRNAs work, or are they created, I should say. But this shows what they do. This is an RNA that no longer is needed. It's made too much of a protein product already. Okay, it's just that it's no longer needed. And so this little microRNA just sits right in the middle of it. If it sits right in the middle of it, the ribosome's not going to be able to come and block the it's not going to be able to come and make the protein. This little thing has effectively set up a roadblock. Okay. At the same time, these little guys, these microRNAs, can be like RNA scissors. They can mark this thing for destruction. Effectively, something will come up and chew up the RNA. Okay, so we can either block the RNA so no ribosomes can actually come translate it, or we can effectively chop up or destroy the RNA so that we can't ever have a ribosome come and attach to it. Okay, so we've effectively, when you think about it, we've effectively regulated gene expression, right? In this case, we've either stopped it temporarily by putting a block there, or we've stopped it completely by chewing up the RNA that makes the protein. We can also block the initiation of translation. Okay, the initiation of translation can be blocked by regulatory proteins. There, I did have a nice animation for this, but it's not overly complicated. It's very similar to how the microRNAs block translation here. Okay, and just in this case, it's done with a regulatory protein instead of a little chunk of RNA. Okay, so basically the same thing. The last way that we can regulate post-transcriptionally is by looking at protein processing. Now, again, this is not necessarily, when I say regulation, a lot of times your minds are going to go right to turning on a gene or turning off a gene, and that's obviously a regulatory mechanism. But in this case, we can regulate protein processing. So let's say we've got our mRNA, we've turned the gene on, we've got the mRNA, we've made the protein. Okay, great. We've turned that process on. But let's say we want to do something a little bit subtle. We want to fine tune. Again, this is a method of fine tuning regulation. Okay, we want to fine tune the final product. We're going to add a few things. We're going to cut it a very certain way. We're going to fold it in just a certain way that allows it to have a very specific function. That's not regulation in terms of turning on or shutting off. That's regulation in terms of making sure we get exactly what we want. Okay. So you can add certain chemical groups, you can send it to a very specific place, you can fold it in a very specific way or cut it in a very specific way. So this protein processing is a point at which we can regulate gene expression as well. All right, and this last one here, as I move this thing up here, um, is degradation of proteins. Okay, so this very, very final stage of regulation. We've got all these things. We've done the mRNA, we've made the protein, we've processed the protein, we've added certain things to it, it's gotten to its certain destination. We don't need those proteins forever, not all the time. Okay, there are times when the protein's just not needed anymore. And when the protein's just not needed anymore, sometimes those proteins get in the way and cause problems in other cellular processes. And sometimes the cell just needs the raw materials um, that protein can provide. So these little things called proteasomes, which are basically like giant garbage disposals, um, they will take a protein, they'll mark it for destruction. They use this thing called ubiquitin. Don't worry about what that is for the test or anything, but it marks them for destruction. And this little guy here, this big orange thing, is the proteasome, and it will recognize this labeled protein as something that needs to be taken care of, and it will chop it up and destroy it. And that's what we see over here. And these little protein fragments over here can be utilized for other cellular processes. They can be recycled and turned into something else ultimately. All right. So we did make it through that big chunk of stuff. And I'm going to stop it at this point. This information on the operons is going to be the next order of business for Friday's class. So I'm going to have you guys watch this one that I just made today, and then we'll come back Friday and we'll do this one. All right, see you then.